Hey everybody, this is your boy Abraham. So today I'm quickly going to talk about the HIV and AIDS. So actually HIV, this is a virus that causes, so this is human immunodeficiency, human immunodeficiency virus. So this virus causes this. So this is acquired immunodeficiency syndrome. So today I'm going to start by talking about the stages of HIV cycle or pathophysiology. So the first thing that I'm going to talk about is what we call binding or fusion. First of all, before we go there, let us quickly understand the viral structure of the HIV. So this is a summary of the virus structure. So this is the outer membrane, and then on the outer membrane there are two proteins. So the first one is GP120. So this is very important. Even this GP41, and also inside we have the integrase enzyme, reverse transcriptase enzyme. We have the RNA genome. So RNA genome, and also matrix. Yes. So this is a summary of HIV structure. So what are the stages involved in HIV transmission or pathophysiology? How does HIV get into the body and establish an infection? Yep. So the first stage, we call it binding. So HIV through the GP120, it binds to CD4 receptor and core receptors known as CCR5, CXCR4. So this is very important, and when it comes to management, so there are drugs which work on this pathway. So the first step is binding. So the GP120 so binds to CD4 receptors. So others, they call this attachment. And then after it has attached and fused. So basically, like in simple terms, like what happens is that let's assume these are CD4 receptors, CCR5 receptors, CXCR4, and then this is GP120. So there's attachment, and then there is fusion. Yes. So this will lead to what we call fusion. So there's out now the what you call rest in peace. So which is the reverse transcript enzyme, integrase protease, even RNA genome is able to enter now into the hot cell. Very, very important. This is the key characteristic of viruses. So on their own, viruses, they are unable to replicate. So viruses on their own, they are unable to replicate. Yes, so they need the what you call host cells. Yep. So in this context, we're talking about the human host cells. So after fusion, fusion will lead to the entering now of RNA genome into the hot cell. We also have you can say just rest in peace. So this R stands for reverse transcriptase reverse transcriptase, and then I integrase, and then P protease. So we're going to talk about this later on. And then the next stage, after binding fusion, so binding attachment fusion, so we're talking about a second pathway is what we call reverse transcription. So inside the old cells, the viral RNA is reverse transcribed into complementary complementary DNA by the enzyme reverse transcriptase. So this enzyme reverse transcriptase transcribed the viral RNA into complementary DNA. And now the next stage is what we call integration. The complementary DNA is transported into the cell nucleus, where it integrates into the old cell's genome with the help of the enzyme integrase. This allows the viral DNA to become a permanent part of the host genetic material. So this is very, very, very important. That's why HIV is a very, very deadly disease. Imagine, so the through the activity of an enzyme called integrase enzyme is able to make parts of what we call complementary DNA into part of the host. Yes, parts of the host cell. So this, in this context, we're talking about a human cell. So very, very important. Yes, I'm going to be quick because I want to briefly talk about drugs. Yeah, so transcription and translation. So the first stage is what we call transcription and translation. So the integrated viral DNA is transcribed into RNA by the host cell's machinery. Some of these RNA serves as the genome for the new viral particles, while other RNA is translated into viral proteins. Yes. So there are two things which happens. So after the DNA, the viral DNA has been integrated. So part A is basically into RNA, so creating a new genome which will create a new viral, and then the other part B is translated now into proteins. Proteins are formed now. It's very, very important. And then the first stage is what we call assembly. Very, very important. So we're talking about the first stage, which is assembly. So under assembly, new viral RNA and proteins are assembled into the immature viral particles at the host cell membrane. So they are gathered, like assembled together. And then the last stage, very important, we call it binding and maturation. Binding and maturation. So maturation. So the immature viral particles bud off from the old cell, taking a portion of the cell membrane with them. The enzyme protease then cleaves the viral protein, resulting in mature infection. Right. So yeah, we're going to talk about the function of the viral protease enzyme. So this enzyme basically cleaves or splits the cleaves of the viral protein. So on their own, before the activity of viral protease, these are like, they are non-infectious, infectious, but after they've been cut, split, they become infectious, and then they mature. Yes, and this is how the life cycle, the pathophysiology goes. So quickly, we're going to talk about basically the drugs. Yes, so known as ARVs, very important antiretroviral drugs. So quickly, based on the life cycle or pathophysiology of HIV virus. So the first group, very potent drugs, we call them nucleoside reverse transcription inhibitors. Nucleoside reverse transcriptase inhibitors. So examples, we have zidovudine, we have abacavir, zidovudine AZT, abbreviation abacavir ABC, and then we have lamivudine 3TC, we have emtricitabine, that is FTC. What else? Let me try to remember. So I have tenofovir. So there's two types. There's TDF and TAF. Very, very important. So how do these drugs work? So this group, nucleoside reverse transcription inhibitors mimic the natural nucleotides that HIV uses to synthesize DNA from its RNA template. When incorporated into the viral DNA, they cause premature termination of the DNA chain. So these, they basically act as the chain terminators. So normally the virus uses a nucleotide. So the reverse transcriptase enzyme reverse transcriptase enzyme using nucleotides to form those chain, creating DNA. So these, they act as chain terminators. So instead of putting the nucleotide, for example, zidovudine is put, as a result, there's no formation of nucleotide. So the chain are terminated, and the process of DNA transcription is inhibited. This is how they work. The second group that we're going to talk about, I'm going to be fast. Second group, we're going to talk about non-nucleoside reverse transcriptase inhibitors. Examples of efavirenz, EFV. We have nevirapine, rilpivirine is highly used in the USA. We also have what we call atravirine. So people have been complaining about my handwriting. So let me put atravirine. Yep, as in ETRPV. NVP. Yes, so how do they work? So non-nucleoside reverse transcription inhibitors, they basically bind directly to reverse transcriptase and induce conformational changes, rendering the enzyme non-functional. So they bind directly to reverse transcriptase enzyme, cause what we call conformational changes. Then rendering this enzyme non-functional. This enzyme is unable to add the nucleotide, making it non-functional. So in the process of reverse transcription. The other group which I'm going to talk about is basically the third group is integrase inhibitors. So integrase inhibitors, we have a very good drug, dolutegravir, DTG. We also have leotegravir. We also have elvitegravir. We also have bictegravir. So this drug, they basically inhibit the integrase enzyme, preventing the integration of viral DNA into host genome. So they inhibit integrase enzyme, preventing the integration. Yep. And then the fifth group is called as entry inhibitors. So entry inhibitors, the first group is fusion inhibitors. So examples of fusion inhibitors of enfuvirtide, abbreviated as T20. So these basically fusion inhibitors block the fusion of HIV envelope with the host cells membrane, preventing viral entry. So as we talked about during pathophysiology, that fusion after the GP120 is attached to the CD4 receptors, there's fusion. So that is inhibited by enfuvirtide. And then in the second group is what we call CCR5 antagonists. A good example is Maraviroc, abbreviated as the MVC. So Maraviroc and other CCR5 antagonists block the CCR5 co-receptor on the surface of CD4 T cells, preventing HIV from entering the cells. Yes. So result, those rest in peace enzyme and the genome RNA can't be transferred into the host cells. So rest in peace is reverse transcription inhibitors, integrase protease. Yes. And then the six ones, I think these are very new agents. We call them We call them post-attachment inhibitors. Post-attachment inhibitors. So example, we have ibalizumab, IBA. Post-attachment inhibitors bind to the CD4 receptor after HIV is attached, preventing conformational changes needed for viral entry. So this they bind to CD4. They bind to CD4 receptor. So inhibit conformational changes. Yeah, so let's quickly talk about common side effects. So the nucleoside reverse transcriptase inhibitors, they affect the lipid profile. They can cause hyperlipidemia. Hyperlipidemia. They can increase cholesterol. So I think I'll do a separate video. On the next video, I'm going to do only just side effects, and also opportunistic infection, and then preventive measures. Thank you so much for watching. So subscribe to my channel, tutorials Abraham, and also tutoring and the consultants. Thank you so much. God bless.